During HIV infection, both viruses and microvesicles called exosomes are produced by virus-infected cells. The overall goal of this procedure is to separate exosomes from the virus particles in plasma samples from HIV-infected patients.
This method can help answer key questions in the exosome field, such as what proteins or RNA molecules are present in exosomes produced during HIV infection. Demonstrating the procedure will be Dr. Ming Huang, instructor in the Department of Microbiology, Biochemistry, and Immunology, and a member of the HIV/AIDS group here at Morehouse School of Medicine.
After collecting human blood and isolating the plasma according to the text protocol, add 10 milliliters of 1x-PBS to 10 milliliters of plasma in a 50 milliliter tube. Centrifuge the sample at 10,000 times G and 4 degrees Celsius for 30 minutes to remove cellular debris. With a sterile serological pipette, transfer the cleared plasma supernatant to a clean 25 milliliter ultracentrifuge tube. Next, centrifuge the cleared plasma at 200,000 times G and 4 degrees Celsius for 2 hours to separate large vesicles. Then carefully remove the supernatant and discard in the biohazard waste. Using 1 milliliter of 1x-PBS, resuspend the pellet and incubate at room temperature for 30 minutes, swirling gently to dislodge and separate particles. Add 24 milliliters of PBS to the just resuspended exosome virus solution in a 25 milliliter ultracentrifuge tube.